To my channel. So in this video, we are going to study about virus, Biroid and prion. Now see, according to the Whittaker system, the five kingdom classification which was given by Whittaker, he was not able to give any place to these acellular organisms. This is important. Acellular organisms like virus, Biroid, prions as well as lichen also. Is it clear? So these are your acellular organisms. Again, it is a very important question area. Now you must have heard about the flu which is caused by the virus. Okay, it is because of the virus. That is the reason we call it as flu also. Matlab, you can understand flu is caused by the virus. Now virus did not find any place in the classification because they are not true living. They are not true living. Why we are saying they are not true living? Because the criteria for living is the cellular organization is important. Okay, which we have studied in the living world chapter. In the living world, cellular organization is a defining characteristics of all life form. So if we understand living as those organisms which have a cell structure, then virus don't have cell, so we call them as non-cellular organism or it is acellular organism. And main thing is this ki virus can be crystallized outside. They have inert crystalline structure outside the living cell. This is important. They are the inert crystalline structure outside the cell. Clear? Once they infect the cell, means when they cause the disease in the cell, then they take over the machinery of the host cell to replicate themselves. If they are multiplying and killing the host, it means they can kill the host. It means they are living also. So what would you call virus? Is it living or non-living? So let us discuss the difference between the living and non-living nature of the virus. Now see, what are the living characteristics of virus? Living nature of virus, because it is able to kill. So first of all, number one point we can say it is able to multiply. Okay, right now we saw it is able to multiply its genetic material. Then second reason is it is showing mutations. It shows mutation also, okay. Then it is causing the disease, means it is infective in nature. And host specific. Now, why we define it as non-living? You can say acellular. Why? Non-living nature of a virus. Virus can multiply, but virus never grow. Growth is a feature which they cannot show. Okay? They cannot show the growth feature. Then they can be crystallized outside. This is a again feature which is telling it that it is non-living, because crystallization we cannot do of living. Then third, it is cannot live independently. It has to live inside the cell only. When we say live, it means it's a living nature. But we say it cannot live without the cell. So it means it's a non-living nature. Inability to live without cell. Without cell, it cannot live. Okay. So these are the natures of the virus. We say it is an intermediate between living and non-living. I hope up till here you are able to understand everything. Now see who gave the name virus. Name virus was it means poisonous fluid. Okay, it means poisonous fluid, and this term was given by Pasteur. So Louis Pasteur, he gave the term virus. Understood up till here? Now see, Ivanovsky, another scientist. Okay, but from here another scientist. Ivanovsky, he recognized that certain microbes as causal organisms of the disease, that is tobacco mosaic virus. They are causing mosaic disease in tobacco. So first of all, Ivanovsky observed that the tobacco plant is showing one disease that is in the mosaic pattern. Okay, and he found that it is smaller than the bacteria. Why he found it is smaller than the bacteria? Because it was able to pass through bacteriological filter proof. Means that filter which can hold bacteria, this was able to pass through that also. So they got an idea, it means it is smaller than bacteria, but it is the one which can cause the disease. And why microbes word is used? Because it is microscopic. Then next scientist is Bajernicke. M.W. Bajernicke in 1898, he also repeated the same experiment just like Ivanovsky, and he told that the extract of the infected plant can cause the infection in the healthy plant. Means if you take out the liquid or you are preparing that extract from the diseased plant, and if you apply this extract to the healthy plant, disease will appear. And that is why he called this fluid is infectious fluid. And it is known as contagium vivium fluidium. So he called that fluid as infectious living fluid. He is using word living because ability to cause the disease. Then the third scientist come Stanley. Stanley was the one who was able to crystallize, and it mainly consists of protein. So crystallization, I told you crystallization is a feature of the non-living. So they are inert outside their specific host cell. The specificity also I told you right now, if they are specificity with their host, they will infect the specific host only. So we call them as, it's better to call the virus as obligate parasite. I hope up till here is it clear? Let me explain you once again, just wait. Now see Ivanovsky, what he has done, he has taken one tobacco leaf. Let us assume this is a tobacco leaf, which is showing the mosaic pattern. Okay, this is the disease, mosaic pattern is coming, the disease. So he grind this leaf. He grind this leaf and he made a paste. Okay, he made a paste. And let us assume this is the filter paper. Okay, this is a one beaker and this is a filter paper which I am keeping on it. And whatever paste is this is allowed to pass through this filter paper. So this paste is allowed to pass. So what we are observing, that the liquid is able to pass through this filter also. So liquid is able to pass through this filter. Is it clear? So it means this filter paper which is able to hold bacteria, this is bacteriological proof filter, means it will not allow bacteria to pass. The bacteriological proof filter, from this filtrate is able to pass. Understood beta? From this filtrate is able to pass. And as a result, you are able to find it is smaller than the bacteria. So this is the work done by Ivanovsky, smaller than bacteria. So he told that the mosaic pattern which is made on the tobacco leaf is caused by the microorganism which is smaller than the bacteria. Now, Bajernicke, what he has done, he has taken this extract and he applied this extract to the healthy plant. Okay. He has applied this extract to the healthy plant. Now, when he is applying this extract to the healthy plant, then what he is finding, the disease is appearing. After the application of this extract, he is applying this extract here. Check, he will apply this extract here. Okay, so after application, what is finding? The disease is appearing. Let me draw once again. So this is a healthy leaf. He has taken it. And after applying the extract, let me write here, application of the extract. What you will find? Symptoms are appearing. So what he told? He told this fluid is infectious living fluid. What he told? Infectious living fluid. Means this fluid is causing infectious and it is living. He used the word living. Clear? Now Stanley was the one who was able to crystallize it. So Stanley prepared it. He prepared the crystal of it. And inside this crystal, what he is finding? The crystals what he is prepared. Let us assume this is the crystal he prepared. And the crystals are mainly made up of protein. So they gave this key, yes, the agent is smaller than bacteria. It can cause infectious also. It means it can spread and it can be crystallized also. So this is the work done by these three scientists. So sequence for learning, you can, I'm just giving you learning tips. Remember IBS. Okay. Sequence, remember IBS. Ivanovsky, Bereznicki, Stanley. This is the sequence of the working you should remember. Then you're in the exams, you will not do any mistake. Bereznicki has and Stanley experiment through which we came to know that crystals are made up of protein. Then we find that in addition to protein, they also contain genetic material also. Now genetic material can be either RNA or DNA. It cannot have both. Virus cannot contain both. It will either contain RNA or it will either contain DNA. It means it is having nucleic acid and it is having protein. So it's better to call virus as nucleoprotein. The virus is called as nucleoprotein. Why? Because it's having nucleic acid and it is having protein. And the infection is not because of the protein. Infectious material is genetic material. Genetic material is the one which is able to cause the infection. Please, if they ask the question, causative agent present in the virus, is it protein or it is DNA only or it is RNA only or more than one option correct, then your answer will go more than one option correct because it is DNA only or RNA only. Okay. Why I'm saying only what? Because you know very well both will not be present together. Now the virus which infect plants, okay, because virus can infect plants, they can infect animals, everyone they can infect, even they infect bacteria also. The virus which infect plants, they have generally single strand RNA. The plant, the one which is infected by the virus which is infecting plant, they will have single strand RNA. Learn this beta, okay? And the virus which infect animals may have either single or double strand RNA or double strand DNA. Anything can be there. So for animal virus, you have to write. For animal, it can be single strand RNA or it can be double strand RNA or it can be double strand DNA. There is no single strand DNA. Please remember, RNA can be single strand. In virus case, the one which is infecting animal, it can be double strand RNA, it can be double strand DNA also. Now the virus which infects bacteria, bacterial virus are also known as bacteriophage. So bacteriophages are the virus which infect bacteria and they are double stranded DNA only. So bacterial virus, for bacteria you have to remember only this. So this is the one which you have to remember for your exam. Ki plant virus are what? Animal viruses are having what genetic material and bacterial virus is having what genetic material. Up till your concept is clear, beta? Now see, the genetic material concept is clear. Now protein. Protein coat is known as capsid and the subunit is known as capsomere. The protein is the one which is known as capsid and it is going to protect the nucleic acid. The nucleic acid is present inside. Clear? Now these capsids which are having the subunit as capsomere, the capsomere can be helical, it can be polyhedral, different forms can be there. So I hope up till here your concept is clear. Let me explain you one diagram. This is the diagram of tobacco mosaic virus, okay? This is tobacco mosaic virus. So this is the one which was first discovered. If you see, this is a capsid An individual is known as capsomere. So these are your capsomere, and see here, this is your single-stranded RNA. Okay, the single-stranded RNA is present in the center, which is surrounded by the capsid. And this is the bacteriophage, means the virus which is going to attack bacteria. Try to learn the diagram carefully. This is known as head, which is polyhedral capsid. Okay, this is short collar. This is sheet, tail sheet. This is your tail fiber, and these are your tail pins. Let me explain you once again. Now see, the TMV, tobacco mosaic virus, length is 300 nanometer, diameter is 18 nanometer, and its molecular weight is 39.4 into 10 raised to the power 6 Dalton. Is it clear? Now see, this is the genetic material, single-stranded RNA. And RNA, if you know very well, let me write here, RNA, which is single strand, it is having nearly 6400 nucleotides. These many nucleotides are present in it. Now this will be surrounded by the protein coat, that which is known as capsid, which is having capsomere. So I'm drawing it one by one capsomere. Try to understand this diagram. This is your one capsomere, 2, 3, 4, this way. Okay, so this is the way capsomere formation will be going on. So this is the way capsomere formation will be going on. And nearly to make the capsid, capsomere are arranged to make the capsid, and nearly 2130 capsomeres will be there. So this is your again important question. How many capsomere are there? Capsomere are nearly 2130. Clear? Let me highlight once again. Please, this is your genetic material. This is your genetic material. Please highlight it. This is your genetic material. Okay. Genetic material is present, which is having 6400 nucleotide, and capsomere is having 2130. Understood? So if they ask the ratio between the nucleotides and capsomere, your ratio will go round figure. You will say the ratio between the nucleotide and capsomere will be 3:1. So between the nucleotide and capsomere, ratio will come 3:1. I hope this topic is clear to you. In the diagram of bacteriophage, see, this is the protein coat. Okay, and this is your short, it will be here neck, here it will be collar. Then you will be finding this is your tail sheet. These are your tail pins, 1, 2, 3, 4, 5, 6 you have to draw. This is tail plate is there, this is tail plate, and the 6 fibers, 3, 4, 5, 6. Okay, and here it will be your hollow sheet will be present inside it. The tail sheets will be this way. This is your tail sheet. And this is the genetic material which is present inside it, because the genetic material has to pass through this area, and when it is infecting, it is passing from here and it will attach to the enter inside the bacteria. Let us assume this is bacteria. Okay, so genetic material has to enter inside this. So this is the way, that is why this hollow core is made in the center, so that the genetic material can pass. So through the tail pins, they will attach to the bacteria, and tail fiber will be there, which is helping in resting. So this is the structure of the bacteriophage. Okay, let me do the labeling. This is head. This is short collar. This is neck, tail sheet, This is your tail fiber. These 6 are your tail pins. Let me write it here. Tail pins. And this one, okay, let me just highlight with another color. This is your tail plate, okay, beta? This is your tail plate. So I hope this diagram will help you to learn and how the working will be there. It is working like a hypodermal syringe only. It will work like a syringe because sheet will contract so that the entire genetic material can pass. This is the way how they infect the bacteria. Now see the list of the disease you have to learn. Diseases: mumps is caused, smallpox, herpes, influenza, AIDS. These are the disease caused in the animals, and in the plants the symptoms. Symptoms is mosaic formation. Number 1, mosaic formation. Number 2, leaf rolling. Number 3, leaf curling. Number 4, yellowing and vein clearing. And number 5 is dwarfism and stunted growth. So I hope this much topic of virus is clear to you. Now let us discuss another sub-viral agent. Okay. Now let us see. Now see Biroid. Biroid was discovered by Theodiner and it is found to be smaller than the virus. And it is causing potato spindle tuber disease. Please learn the short form also, PSTD. And it is nothing free RNA. It is not having any protein coat, no protein coat. That is why the name is given Biroid, and the RNA is also of low molecular weight. It is not of high molecular weight. Clear? It is of low molecular weight. Another agent is prions.